Hello students, hope so you are doing well, and in our today's lecture we will discuss about the exercise of chapter 3, that is enzymes. So let's start with the fill in the blanks. Try to solve the puzzles or fill in the blanks. First pause the video and after that play and see the answers. Enzymes are composed of hundreds of amino acids. Some enzyme consist of a non-protein part known as a cofactor. Many enzymes require a non-protein part component called the cofactor for their proper functioning. Enzymes are highly specific in nature. Enzymes which carry out the synthesis of proteins are the integral parts of ribosome. So next is the true false. The enzyme important in photosynthesis are found in the entire mitochondria. Large amount of enzyme can accelerate chemical reactions. No, only small amount. And the enzymes that are present in photosynthesis are found in chloroplast. So both of them are false. Calvin Melvin proposed lock and key model. No. Emil Mixture. The active site of enzyme is composed of 4 regions. No, 2 regions. That one is a binding site and another is catalytic site. Structure of enzyme has no specific importance. It is also false. Structure of enzyme has a specific importance. The next is short questions. List 2 conditions that destroy the enzymatic activity by disrupting bonds between the atoms in an enzyme. So first of all, there is a temperature. If we change the temperature, then after a certain limit, the bonds of the enzyme will denature. And the other factor is the extreme change in the pH. Not only pH, the extreme change in pH disturbs the bonding of enzymes. Question 2. How do low and high temperature affect an enzyme activity? If temperature is low, the collisions between the molecule will be low, or we can say that the thermal energy will be low and the rate of reaction will be low. And if we increase the temperature, of course there will be more collisions, there will be more kinetic energy, and there will be more reactants are changed into products, and the rate of reaction will be more. Next, question number 3 is what is a prosthetic group? As we have discussed in our lecture, prosthetic group actually, what is prosthetic group? Actually, prosthetic group is a non-protein part, but it is attached covalently with the protein part. If non-protein part is attached loosely, and if it is organic and if it is inorganic. If it is organic, it will be the coenzyme and like NAD, and if it is inorganic, it is called the activator, and for example magnesium, copper and all the metal ions. So define inhibitor of the enzyme. Question 4. Inhibitors are actually the substances that inhibit the action of enzymes, that are poisons like cyanides and different antibiotics. There are 2 types of inhibitors, reversible and irreversible. How does an enzyme accelerate a metabolic reaction? By lowering the activation energy. As we know, the activation energy is the amount of energy, least amount of energy that is required to change a reactant into a product or to start or initiate the reaction. So by lowering the activation energy, actually when we use enzyme in any chemical reaction, what actually enzyme do? They lower the activation energy and it is quickly achieved, and the reaction and the reactants are changed into the products. Thank you. This was the exercise, and in our next lecture we will discuss and we will start the chapter number 4, that is cell. Thank you.